have you ever just stopped and wondered how the invisible abstract code that's tucked inside every single one of your cells becomes, well, you? You know, your hair, your muscles, even the enzymes digesting your lunch right now. It's kind of mind-blowing. Today we're diving deep into one of the most fundamental processes of life, the story of how that genetic information becomes physical reality. It's a process called translation.
And really, that's the big question, isn't it? How does this happen? Deep inside your trillions of cells, there's this microscopic drama unfolding, constantly, turning a simple four-letter alphabet into the incredibly complex machinery of a living, breathing person. This process, it's the bridge between the blueprint of life and life itself.
So let's break it down. The core mission of your cells is actually pretty straightforward. Every cell has a blueprint, right? A working copy of its genetic instructions, which is called messenger RNA, or just mRNA for short. This little blueprint has all the info needed to build pretty much everything. And the process of reading that blueprint and actually building the thing, that's translation.
And the scale of this whole operation it's just staggering. I mean, we're talking about tens of thousands of different kinds of proteins. You've got proteins for structure, for transport, enzymes that make chemical reactions happen, all of them built by this one single elegant process. And get this, it's happening inside you right now, billions and billions of times over.
Okay, so to really get how this works, let's picture it like a tiny molecular factory. We've got a blueprint to read, we've got raw materials that need to be assembled, and of course, we need a machine to do all the work. So let's go meet the key players on this amazing cellular assembly line.
The factory itself is this incredible machine called the ribosome. Now, it's actually made of two separate parts, a large piece and a small piece, a large and small subunit. Most of the time, they're just floating around separately. But when it's time to build a protein, bam, they come together and clamp right down onto that mRNA blueprint, ready to get to work.
Of course, no factory could run without raw materials. For us, the building blocks are these molecules called amino acids. And what's wild is that there are only 20 different kinds of them. That's it. The whole incredible diversity of life is built from just 20 building blocks. The specific sequence of these amino acids is what gives every single protein its unique job.
So you might be asking, how do the right amino acids get to the factory at exactly the right time? Well, that's the job of this little guy, transfer RNA, or tRNA. The best way to think of it is like a specialized delivery truck. Its whole purpose is to read the blueprint and go grab the one specific amino acid that's being called for. It's the perfect adapter molecule.
And this brings us right to the heart of the whole process, actually reading the code. The language written on that mRNA blueprint is surprisingly simple. It's written in words that are only 3 letters long. So let's see how the factory deciphers this.
The system here is just beautiful in its simplicity. Each 3-letter instruction on the mRNA blueprint is called a codon, and our little tRNA delivery truck, it has a matching 3-letter sequence called an anticodon. When the codon and anticodon match up, it's a perfect fit, like a key in a lock, and that's how the ribosome knows the correct amino acid has just been delivered. Now, inside our ribosome factory, there are 3 main workstations, or sites. You've got the A site, which is the arrival dock where the new tRNA comes in. Then there's the P site, the main processing station where the growing protein chain is held. And finally, the E site, which is the exit, where the used empty tRNA gets kicked out. So just remember, A, P, E. Arrival, processing, exit. Simple as that.
All right, we've met the crew, we've seen the factory floor. Now for the main event. Let's put this assembly line into motion and actually watch a protein get built, one amino acid at a time. Okay, here we go. The cycle kicks off. A new tRNA, charged up with its amino acid, zooms in and docks in that A site, perfectly matching the codon on the mRNA. And right next door, in the P site, is the protein chain that we've been building so far, just waiting. And here it is. This is the magic moment. The ribosome itself is actually a giant enzyme. It reaches over and forges a super strong bond, called a peptide bond, that attaches the brand new amino acid to the end of the growing chain. And just like that, the protein is 1 unit longer. So what happens next? The entire ribosome machine just scoots over one codon down the mRNA blueprint. This one move shuffles everything over. The tRNA that was just holding the chain is now empty, so it moves to the E site and gets ejected. And the tRNA with our newly lengthened protein chain, it slides right into the P site, leaving the A site wide open for the next delivery. the cycle is ready to go again.
This goes on, adding amino acid after amino acid, chugging along the mRNA until the ribosome hits a stop codon on the blueprint. Now, this doesn't call for another tRNA. Instead, a special protein called a release factor comes in and binds to the A site. It basically acts like a pair of molecular scissors, snipping the finished chain of amino acids, which is called a polypeptide, free from the ribosome. And with that, production is complete.
But wait, we're not quite done. A freshly made polypeptide is just a long, floppy string of amino acids. It's not a functional protein yet, not by a long shot. To actually become useful, it needs to be folded up, finished, and sometimes even modified.
You see, for a protein, shape is absolutely everything. And it gets that shape in a few stages. The primary structure is just that sequence of amino acids we built, like a string of beads. Then it starts twisting and folding into local shapes, like coils and sheets. That's the secondary structure. All of those then fold up on each other to create the final complex 3D shape, the tertiary structure. And sometimes, several of these folded chains have to come together to form one giant protein machine. That's quaternary structure.
And the finishing touches don't even stop there. Take insulin, for example, the hormone that regulates your blood sugar. It starts out as a single inactive polypeptide. Then it gets shipped out, folded up, and has entire sections of it literally snipped out before it becomes the final active hormone. This post-translational processing is a super critical final step for a lot of proteins. And here's the thing that's just so profound. This whole process, the ribosome factory, the tRNA delivery trucks, the three-letter code, it's basically the exact same in every single living thing on this planet. A bacterium, a mushroom, a blue whale, us. It's all the same system. This universality is some of the most powerful evidence we have that all life descends from a single common ancestor. The same molecular machinery that builds a tiny microbe is building you right now. Translation isn't just a cellular process. It's the shared universal language of life itself. Just think about that for a second. What does it mean that all of life shares one common creative tongue?